Okay, and so now we will go into the topic of genetic inheritance, and I will start with the subtopic of 4.1 about the Mendelian genetics. And I will start with some terminologies and the introduction to this Mendelian genetics. Firstly, what is a gene? So a gene is actually a specific sequence of nucleotides, okay, that can be found along this long DNA molecule, okay, which is actually part of our chromosome. And this gene codes for a specific protein, which later determines a certain characteristic that can be passed on from one generation to another generation. And so this gene is considered as a unit of inheritance for a particular characteristic. Okay, for example, in a plant, there will be this certain color for the flower. So the factor that will determine the color of the flower will be a gene known as the gene for flower color. Okay, and all these genes will be located on a chromosome. So the position or the location of the gene on a chromosome is known as a locus. Okay, referring to the word location. So location, position of the gene on a chromosome, that will be a locus. And of course, there will be many genes on this one chromosome, and so there will be many loci, okay, for this one chromosome. So locus is the singular form, meanwhile loci will be the plural form of this word. Okay, so if you can see in this the diagram, this will be maybe this an example as the locus for this particular gene, the gene for the color of the flower. And usually a gene will be having two alternative forms, usually, maybe more. Okay, and so an allele is referring to the alternative form of a gene. For example, let's say we are talking about the gene for flower color. Usually this gene exists in two forms or two types. So if the gene is controlling or determining the purple color for the flower, then we can call that as the allele for the purple flower. If another form of that same gene will determine the white color for the flower, then we call it as the allele for the white flower. So allele and genes are actually the same. Okay, it's just the allele will be referring to the maybe two types of one particular gene. Okay, so as you can see in this diagram, this will be two homologous chromosomes. Both homologous chromosomes are carrying the same type of gene. Let's say the gene for flower color, but there will be two forms of this same gene, in which one will be the allele that will be giving the color purple to the flower. Another one will be the allele that will be giving the white color to the flower. Okay. If one individual has got two homologous chromosomes, two chromosomes that are homologous to each other, and we should know that one of the chromosome comes from the maternal side or the mother. Let's say this one. Okay, let's say this chromosome comes from the mother, and this other chromosome will come from the father or the paternal side. So usually in one individual there will be chromosomes that are homologous to each other. Okay, and if for a particular gene there will be two identical alleles, such as this one, capital A and capital A alleles. Okay, so we call it as a homozygous condition. So homozygous condition referring to a condition in which one individual is having two identical alleles for a certain gene. Okay, so if there are two alleles that are similar, capital A, then homozygous. The same goes for a condition where one of the homologous chromosome is having this small a allele, another one will also be having the small a allele. We also say that this is showing a homozygous condition. Okay, and if this allele is a dominant allele, Okay, then we can say that this condition over here will be showing the dominant homozygous condition. Meanwhile, if there are two small alleles, okay, over here, we can say that this will be referring to the recessive allele, and so this type of condition is known as the recessive homozygous condition. Okay, so dominant homozygous is a condition in which one individual will be having two dominant alleles, the same one. Meanwhile, recessive homozygous is a condition where one individual will be having the same recessive alleles, small a and small a. Meanwhile, heterozygous is a condition in which one individual is having two different type of alleles for one same gene. So the gene is one same type, but there are two forms of the gene. One will be capital A and another one will be small letter a. So if there are two types of alleles, two different types of alleles for one same gene, then we call this as the heterozygous condition. Okay, so just now I've mentioned about the word dominant allele and recessive allele. So what is actually the dominant allele and recessive allele? We will start with this one. A dominant allele is an allele that can be expressed in homozygous condition or heterozygous condition. Homozygous referring to, for example, the same type of alleles just now, capital A and capital A. While heterozygous referring to the condition in which there are two different types of alleles, capital A and small letter a. For example, you can see in this diagram, this will be showing the homozygous condition, and this will be referring to the heterozygous condition. So, for example, the purple flower allele in plants is said to be dominant. Okay, dominant. Why? Because this particular allele can determine the purple color to the flower, even though it is in a heterozygous condition, in which even though there is only one of this allele, it is enough for this allele to show its characteristic. Okay, and usually for the dominant allele, we use the capital letter as the symbol. So for example, we use the capital letter P for this allele for the purple flower. Meanwhile, recessive allele will be referring to allele that can be expressed only in homozygous condition. For example, like this one. Okay, we can see that there will be these two recessive alleles. Okay, small letter A and small letter A. And for example, in the plants, the white flower allele is said to be the recessive type of allele. Okay, in which this allele can only produce white flower only if there are two of this allele, and usually we will use the symbol using the small letter. And usually we will use the small letter for the symbol for this recessive allele. So for example, we are using this small letter P. Okay, and so the plant will be having white flower only if there will be these two small letter P alleles together. Okay, and the next term that we need to know will be the term of genotype, in which this refers to the genetic constitution. The genetic constitution, ataupun we can also say that this refers to the genetic composition for a certain character of an organism. This will be different from another term known as the phenotype, which refers to the physical appearance for a certain character of an organism, something that we can see normally, something that is observable. For example, let's say there is this allele for red color, and this allele is a dominant allele. So we use the symbol of capital letter R. Another allele will be the allele for white color, and this allele is recessive. So we use the small letter R as the symbol. So if this one individual plant has got two of these capital R and capital R alleles, so the genotype will be capital R, capital R. Okay, So this shows that this individual has got these two alleles, and this becomes the genetic composition that will give the characteristic of red color. And so since red color is something that is that can be observed, so we say that this is the phenotype for this individual. So genotype will be referring to the types of alleles that can give the effect on the characteristic of this individual. Meanwhile, the phenotype will be the appearance that we can observe. Okay, for example, red color. So what if the individual plant has got this capital R and also the small letter R alleles, the dominant allele and the recessive allele together in a heterozygous condition? So the genotype will be capital R and small letter R. Okay, showing a heterozygous condition, and red color will be the phenotype. Okay, due to this dominant allele over here, the capital R. Meanwhile, if this individual plant has got two small letter R alleles, okay, so we can see that the genotype will be small r and small r, or the recessive homozygous condition, and the phenotype will be white. Okay, because there is no capital R allele, there is no dominant allele, so the recessive allele will be able to show its characteristic of white color. Okay, so please differentiate between the genotype and the phenotype. Okay, in a genetic study, usually we will do a crossing, kacukkan. We will do a crossing between two individuals that is considered as the parental generation. The parents individuals will be part of the parental generation. So we can use the symbol P, okay, to refer to the parents generation. And so let's say we have one plant that is having purple flower, and we cross this plant with another plant that will be having white flower. As a result from this crossing, the offspring will be produced, generasi anak, the offspring, and this offspring is known as the F1 generation, or the first filial generation. So this F1 generation will be showing purple flowers, all of them. All of the F1 generation will be having purple flowers only, no white flowers whatsoever. Okay, but in genetic studies, usually we will continue to do another cross, in which we will take one individual from this generation and we will cross with another individual of the same generation. So what we are doing actually will be this self-cross. This is a special type of crossing in which we call it as the self-cross. So what is a self-cross? A self-cross is a cross that involves individuals of the same generation. As you can see, we cross individuals from this F1 generation. So the symbol will be F1 crossed with F1. Okay, and as a result from this self-cross, we will get the F2 generation, the second filial generation. And for this F2 generation, most of the plants will be having purple flowers. Most, okay, many of them will be having purple flowers, but there will be a few, and there will be some that will be having white flowers. So this is something unique for the F2 generation. Okay, but what is important right now will be the term of self-cross that we need to know right now. Another example will be this crossing involve a plant that is tall, and we will start with a homozygous dominant plant, so capital T, and we cross this particular individual with another individual that is homozygous recessive plant, which is short plant. Okay, a short plant, and the genotype will be small t. So these two will be the parents of this crossing, and so this is considered as the parental generation. From these two parent individuals, we should know that this will be the gametes that can be produced. The gametes. So from this individual, there will be this one type of gamete that will be containing this capital T allele. Meanwhile, from this other parent, this gamete will be containing the small t in it. And from this crossing between these two gametes, we will be producing the F1 generation that will be having this type of genotype, capital T, small t, the heterozygous condition. And the phenotype of these F1 individuals will be tall. And we will continue with the self-cross F1, cross with another F1 individuals. This will be the genotype, of course, the same like this particular F1 generation. And the gametes from this heterozygous individual will be these two types, in which one of the types of gamete will be having this capital T allele. Meanwhile, another type of gamete will be having this small letter t allele. The same goes for this individual, the same type of gametes, capital T and small letter t. So if we cross these gametes together like this, we will be getting the F2 generation, in which this will be the genotypes of the F2 generation. Okay, there will be one type showing the homozygous dominant condition, capital T, which will be tall plant, and there will be two out of 4 that will be showing heterozygous condition, capital T and small letter t, and there will also be tall plants because of the dominant allele. And there will be 1 out of 4 of the F generation will be showing a recessive homozygous condition, small t, in which the phenotype will be short plant. Okay, so in this diagram, okay, we can see the self-cross that is being done on the F1 generation individuals. Okay, there is another term that we need to know, and that will be the term known as test cross. So what is a test cross? A test cross is the mating of an individual with a dominant phenotype but with unknown genotype to a homozygous recessive individual. Okay, so why do we need to do this test cross? This is to determine the unknown genotype of this dominant individual. Okay, for example, let's say we see a tall plant. Okay, a tall plant. So this tall plant is showing a dominant phenotype. Okay, tall is the dominant phenotype. However, we are not sure about the genotype of this individual. Okay, is it homozygous dominant, capital T, or maybe heterozygous, capital T and small letter t? So we are not sure, because both of the genotypes can still produce this type of dominant phenotype. Both genotypes, T besar, T kecil, boleh menghasilkan phenotype sama, iaitu pokok yang tinggi. Okay, and so we will do a test cross. Just to determine this unknown genotype over here. Okay, we know that there will be one capital T, but what about the other allele? We are not sure about this. So we do a test cross. How? By crossing this individual with a homozygous recessive individual, in which we will cross this tall plant with a short plant or dwarf plant. Why? Because we can confirm that this individual will be having this homozygous recessive genotype, small t. Okay? And how can we determine this unknown genotype? Okay, we can determine this by observing the offspring, the result of the test cross. So when we do this test cross, we will get the offspring, generasi anak. So we look at the offspring, we observe the offspring, and if all of the offspring are tall plants, then we can conclude that the genotype of this tall parent is homozygous dominant, capital T. Okay, if 50% of the offspring are tall plants and another 50% are dwarf plants ataupun short plants, then we can conclude that the genotype over here for this tall parent will be heterozygous, capital T, small t. Okay, how do we explain this? Okay, this can be explained by this genetic diagram, okay, in which we can see that if all of the offspring from the test cross are tall plants, then we should know that this genotype will be homozygous dominant, capital T, capital T. Okay, but if 50% of the offspring of the test cross are tall plants and another 50% of the offspring are short plants, then we can conclude that this genotype will be heterozygous condition, in which it will be capital T and small letter t. Okay, so this genetic diagram help us to understand, okay, and determine the genotype of this tall plant. Okay, so this is about the test cross, okay, that can be done to determine a particular unknown genotype. And so now we will look at the introduction to the Mendelian genetics. So what is actually this term referred as the Mendelian genetics? So this refers to the basic principles of heredity that was proposed by a scientist named Gregor Mendel. And Gregor Mendel performed a lot of genetic crosses using a special type of plant known as the garden pea plants. The scientific name is Pisum sativum. Okay, why Mendel used this particular garden pea plants? Why not other type of plants? Or why not other types of organism? Animals maybe, humans maybe. Why not? Okay, so Mendel used these garden pea plants because this type of plant is grown easily in large numbers. Other than that, the plants will be having short life cycle and their pollination could be controlled. And there will be seven easily observable characteristics for Mendel to observe in his experiments. So this table shows the 7 observable characteristics of Mendel's pea plants. So you need to know that sometimes Mendel will look at the length of the stem, ketinggian pokok, in which the pea plant can either be tall plant or short plant only. No in between, no intermediate, either tall or dwarf only. Another characteristic will be about the shape of the pod, either inflated, gembung, or constricted. And another one will be about the shape of the seed, in which some will be round in shape and some will be wrinkled. And next will be about the color of the seed. Some will be yellow and some will be green in color. Another characteristic that can be observed will be about the position of the flower, either axial at the axis of the stem or the terminal position at the end of a particular branch. And another characteristic will be about the color of the flower. I mentioned just now, maybe the color of the flower will be purple or white. And the 7th observable characteristic will be about the color of the pod, either green or yellow.